Hello, everyone. I'm Xinwang Yu. I'm a research associate from NC State Tufgrass breeding program. Um, my research focuses on developing genomic information for tufgrass, which could help us to understanding the genetic control of a tufgrass respond to the environment stress, and how this genomic information could help us improve the breeding efficiency. Today, I will introduce our studies together with graduate students Sydney and Curtis. We all know the conventional breeding used on the tufgrass is based on selection of the target phenotype. Alternatively, the molecular breeding usually select the DNA markers that tightly linked to the phenotypic traits. We call this marker-assisted selection. The advantages of marker-assisted selection include the early selection in the very early seedling stage, and the reduced cost and cycle time, and screening more efficient. However, the application of marker-assisted selection on the breeding will be based on a precondition that the DNA markers can reliably predict the phenotype. So in the first step, we need to identify the DNA markers that are associated with the target trait. We have been conducting our research on the Augustinegrass and the zoysia grass. In Augustinegrass, our breeding goal is to improve the drought tolerance, freezing tolerance, and the gray leaf spot resistance. In zoysia grass, we try to improve the freezing tolerance and the large pad disease resistance. Okay, let's look at the first project, the drought tolerance on the Augustinegrass. We choose the Raleigh and the Seville and the parent lines and cross the resulting in 115 hybrids. We tested the drought tolerance of the population in both greenhouse and the field test. We could see after the drought stress, some plants have been turned into yellow and wilt, while someone still looks green. These slides show the result of QTL mapping for drought tolerance. We identified several genomic regions that controlling the drought tolerance, especially the one region on the group of R6. In this region, several genomic regions could be repeatedly detected in different trees and different environments, which we believe it has a great value to be used in breeding selection. The next project is the gray leaf spot resistance in Augustinegrass. The two parent lines, Raleigh is the relatively gray leaf spot susceptible to the disease, while the plant introduction PI 410353 is relatively resistant to the gray leaf spot. The cross of the two parents results in 158 F1 hybrids. We tested the disease resistance of this population in the phytotron, and the population showed the high segregating for the gray leaf spot resistance. After the genetic analysis, we identified several genomic regions controlling the gray leaf spot resistance, especially these two showed on this slide on the group P2 and the group P5. they have the high peak for the association between the markers and the grey leaf spot resistance. We believe the marker in these two regions have the potential to be used in breeding selection. In the next project, I will invite Sydney to introduce her research on the freeze tolerance in Augustinegrass.
Hello, everyone. My name is Sydney Graham. I am a master's student in Susanna's lab, and my projects are mostly on the genetic improvement of St. Augustinegrass for freezing tolerance. So just a little bit about this project. We have two different populations for this project, and we are using both of them for mapping. One of the projects is Raleigh by Seville, which is the same population that Shimon was telling you about for the drought evaluation. So this was tested in the field and in freezing tests. And we also have a Raleigh by Raleigh population. We have a similarly sized population here, which was screened in freezing tests. And in this photo here, you can see some of the variation in response to freezing in St. Augustinegrass. And then this is from a publication by Kimball et al. From 2018. And it shows some of the genomic regions that were identified to be associated with freezing. As you can see, there's quite a few regions on linkage groups 3 and 9. And we are working on updating these results and verifying them using the Raleigh by Raleigh mapping population. And we should have updated results and more narrow genetic regions coming soon. Thanks.
Okay, thanks, Sydney. Okay, let's move to the zoysia grass. In zoysia grass, we try to identify the markers associated with the freezing tolerance. We use the Meyer and the Victor and the parent line to cross the mapping population. Meyer is the good co-tolerance, while the Victor is less co-tolerance. The mapping population containing 175 progeny. The population was first tested in the 3 locations in Georgia, North Carolina, and Indiana. We also tested the freeze tolerance of the population in the controlled freezing test using the growth chamber and the freezer. These slides show the result of QT mapping for both field test and freezer test. The different color of the bar showed here indicate the genomic regions and identified in both field and freezer, which means they have the high potential to be used in marker-assisted breeding selection. And this study was conducted by the graduate students, McKamey and Jessica. Okay, the next project is the luck patch resistance in zoysia grass. We'll transfer to Curtis to introduce his project.
Hi, I'm Curtis Houting, first year graduate student, master's student in Susanna's lab, and I'm doing mostly the mapping of large patch resistance in zoysia grass. So the population that we're going to be mapping, it was gone through some initial screening, and in that screening it was found that PI 231146 had the highest resistance to both. There was 2 strains of the isolates of the disease-causing vector that causes large patch, and we're going to be using that variety, and then also doing a cross with the commercial availability of Meyer. And this will be an F1 screening, And we're going to hopefully use this to figure out different genetic markers that will help to assist with future breeding for genetic resistance for large patch resistance. Can't move. So I already talked about this preliminary screenings already, but we're going to be kind of following the same methods that they use. We'll be using inoculated seeds, and most of the study will be done in a controlled environment inside the growth chambers, where we can control the humidity and keep it at optimum growth for the bacteria to infect and see what ones are truly resistant.
Okay, thanks, Kurt. So we have from the all above projects. In summary, we have established the technologies, including the genotyping and phenotyping, which could be help us to detect the DNA marker and the trait association in warm season grasses. We have identified several DNA markers associated with the desirable traits in Augustinegrass and zoysia grass. However, the effect of these markers still need to be validated before applied in the breeding selection. Okay, thank you for your interest in our research. You are very welcome to contact us if you have any question. Thank you.